Hello, students. Welcome to Chapter 7 and Chapter 9. We're going to talk about brain development, mostly in utero, and then also epigenetics. So these chapters kind of put together quite a few things that are really relevant across the lifespan and across the class. So this might fit better earlier on in the class, might fit really well at the end of class. So depending on the quarter, this may be in a different place in the class. But to begin, let's start with Chapter 7 on brain development. Well, we will start basically at the beginning of the lifespan in the embryonic stage. So the embryonic stage is after the fertilization, after the zygote stage. The embryo is implanted into the uterine wall. So at about 16 days after fertilization, we see the embryo forming 3 layers of cells that basically become the rest of all of the body's organs. So all of these are kind of varieties of stem cells specifically that become all the different cells in your whole body. So the endoderm is not one we'll focus on very much. So endoderm is going to become the inner tissue, a lot of your internal organs, very important, obviously. The mesoderm is the middle tissue here, and it is becoming the muscles and ligaments, so connective pieces of your body. The main thing that we will focus on is the one pointed out in these pictures, and that is the ectoderm. So the ectoderm becomes the outer tissue, but in addition to becoming the outer tissue, like your skin, your hair, your nails, so things on the outside of your body, it also becomes your nervous system. So all of these stem cells that are dividing become all the different organs of your body. So at that 16 days, we see these 3 layers very clearly as distinct pieces. And what happens is this neural fold happens at all because of various complicated, basically molecular moving things around and certain chemicals that are released in the process in the development of a embryo to essentially tell these stem cells to organize in specific ways and then make this neural fold, make this neural tube, then start to divide from that point. So the ectoderm is the main one that we're concerned with here in the neurobiology class because that becomes the nervous system. So I wanted to watch some videos on this. Both of these are pretty good, pretty short, and they give you hopefully a better sense of what is actually happening during this time. So, and they're also linked here if you have to just click on them. If I get a copyright strike again on anything, just click the link in the slides if you don't see it pop up into that video. But we'll do this one first.
Neurulation is the process of neural tube formation and its development into the spinal cord and the brain. Any complication during this complex process can result in neural tube defects. The development of the nervous system begins during the 3rd week of gestation. Primary neurulation is the process that forms the functional central nervous system. Let's begin with the development of the embryonic nervous system. By day 18 of fetal development, the ectodermal germ layer has the shape of a disc with a cranial and caudal end. The cross-section on the right illustrates 3 germ layers: the ectoderm, mesoderm, and endoderm. At this point, the central nodal cord has induced the overlying ectoderm to thicken and form the neural plate. The cross-section on the right will help visualize the folding of the neural plate, leading to the formation of the neural tube. By the end of the 3rd week, the edges of the neural plate extend upward to become neural folds. The depressed mid-region is now the neural groove. Each neural fold then grows toward each other to fuse at the midline, converting the neural groove into a complete enclosed and hollow neural tube. At the same time as the neural folds fuse, a population of cells separates from the tips of the neural folds to become a mass of mesenchymal cells called the neural crest. The neural crest cells migrate throughout the body to form a wide variety of cell types, including Schwann cells, the meninges, endocardial cushions, parafollicular cells, and the adrenal medulla. Before the fusion of the neural tube is complete, the cephalic and caudal ends communicate with the amniotic cavity through the cranial and caudal neural pores. The cranial neural pore closes on day 25. After closure, the cranial end of the neural tube develops into three primary vesicles: the forebrain, or prosencephalon, the midbrain, or mesencephalon, and the hindbrain, or rhombencephalon. These three develop later into five secondary vesicles. The forebrain develops into the telencephalon and diencephalon. The midbrain develops into the mesencephalon. The hindbrain develops into metencephalon and the myelencephalon. The caudal neural pore then closes on day 27.
The rest of that video is pretty interesting, but we will jump to the next video from 2-Minute Neuroscience. It does kind of cover some of what was just covered in that first video, but it goes a little bit beyond there. So what are the divisions of those different of the pieces of the nervous system?
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss early neural development. The development of the nervous system begins at around the 3rd week of embryonic development, when an area of the ectoderm, or the outer tissue layer of the embryo, thickens and forms what is known as the neural plate. This plate begins to fold inward, forming a groove called the neural groove. The sides of the neural groove, known as the neural folds, begin to come together. At the end of the 3rd week, the folds will begin to fuse together. By the end of the 4th week, they have completely fused together to form the neural tube, which will eventually become the brain and the spinal cord. As the neural tube closes, bulges and bends begin to appear and they gradually become more noticeable. During the 4th week, there are 3 of these bulges present. They are called the primary vesicles. They are the prosencephalon, which will eventually form the cerebrum, the mesencephalon, which will eventually become the midbrain, and the rhombencephalon, which will eventually become the rest of the brainstem and the cerebellum. The end of the neural tube will form the spinal cord. As the brain continues to develop, 2 of these vesicles further subdivide to form secondary vesicles. The prosencephalon forms the telencephalon and the diencephalon. The telencephalon will become the cerebral hemispheres. The diencephalon will eventually consist of the thalamus, hypothalamus, and other structures. The mesencephalon does not subdivide any further and will become the midbrain. The rhombencephalon will subdivide into the metencephalon and the myelencephalon. The metencephalon will become the pons and the cerebellum, and the myencephalon will become the medulla. The spinal cord will still form from the end of the neural tube. Next, the neural tube will continue to develop to look more like the brain. The telencephalon will grow more rapidly than other parts of the tube, and by 11 weeks, the brain will have a similar shape as to what it will at birth. Although it will continue to develop after birth, the brain at birth is structurally similar to a fully developed brain.
So one main thing that I hope you take away from that with the animations, hopefully it's a little easier to understand, but basically in that first month, we have a lot of processes happening to start the development of the brain. Just like all the other organs in the body, as we're in the embryo stage, it's basically all of the organs in the body starting to organize, moving where they're going to be, and that's what's happening with the brain as well. So that endoderm folds up, becomes that neural fold, becomes that neural tube, and that continues to become more what we kind of expect to see from a brain. So again, all of these processes from the first 2 videos here, getting to that neural tube, that basically takes that first month, about 4 weeks total. And then one thing that we see is that after that neural tube is formed, we start to see neurogenesis. So neurogenesis is just fancy word for making neurons. So essentially a neural stem cell becomes neurons. So, and neural stem cells can divide, as we've talked about before. Neurons themselves, like fully developed neurons, cannot divide. You don't have neurons in your brain doing cell division to make new neurons. You can only get that from neural stem cells. So the stem cells, they're the special ones that can divide, but they can't do the communication piece. So a neural stem cell doesn't have like axons and dendrites yet. but they have the ability to divide and then become a fully developed neuron. So if you think of it like a Pokemon, it's the first stage. It's a neural stem cell is the baby stage of a Pokemon, and then a first evolution is the fully developed neuron or something like that. If that example didn't work for you, totally fine. All right, so with that, we can see here, I think I've got in another couple slides. Yep, I'm going to jump to this slide so we can see this a little bit bigger. So you can see here the neural tube here at about the end of that point, so about 4 weeks, and you can see it folding up to look more and more like a brain. And so that is what's happening there is that at the same time, once you get that neural tube formed, essentially you're getting instructions, hey, all you neural stem cells, divide, make tons more neural stem cells, and then also migrate to where you're going to live the rest of your life, right? So some of these neural stem cells are going to end up part of your prefrontal cortex, part of them are going to be your cerebellum, right? So they're essentially dividing, making tons of them all the way up to not even when you're born, but when you're a young child, about 18 months, like a year and a half after you're born, about 2 years, you might say, you still have a lot of this neurogenesis and moving those neural stem cells to their final resting place, I guess you could say, in the brain. Yeah, so take a look at this image and see what kinds of things you can learn about the brain development over that time. So it's pretty impressive how much division is happening. I mean, in the embryonic stage, there's tons of cell division happening everywhere. But since this is a neurobiology class, we're going to focus on the brain. So about 4.6 million neurons are generated each hour. So during this time. So as that process gets ramped up, so you're in week 6, week 7, week 8, you now have a ton of neurons being developed. So those neural stem cells dividing, and then they will migrate to where they are going to be. So essentially, this neurogenesis continues on even after you're born. So it's still happening until close to 2 years old. So the book has 18 months is like it's not a hard cutoff. Again, this is on average. So some people it'll stop a little sooner, some people will stop a little bit later, but it's basically genetically determined. There's not a lot of environmental influence here other than like brain defects. So there's also things with epigenetics we'll talk about and stress and changing the structure slightly, making them act a little bit differently. But the main structure of the brain is pretty genetically set to go into those formats. So you've got your temporal lobes, your cerebellum, prefrontal cortex, those kinds of structures. So the basic brain structure is in place after the second trimester. It looks pretty similar to what it'll look like when you're born, when you're a young child, when you're an adult. Again, most of our neurogenesis is happening in that early time. So as you can see here, I believe some of these I think are actual pictures of brains. Some of them are a little bit artistic rendering. But the brain looks pretty similar at birth as it does the rest of our life because we've got most of the neurogenesis, most of the neurons to where they're going to be forever. All right, so what happens as that neural stem cell divides? Essentially, it's not in its final location. It might have to move all the way to the prefrontal cortex, all the way to the back of the brain. So essentially, we have these neural stem cells as they are a stem cell and becoming a full neuron as they're making those chemical processes, they move. They move to their final locations. There's other brain cells that we'll talk about in a little bit that help with that movement and chemical processes, help guide it to where it's going to go and other things, other structures, amino acids, proteins, all sorts of complex things that your book goes into a little bit of detail, but essentially neural stem cell divides, you've got more neural stem cells. As it's moving, it's becoming an axon, or sorry, it's becoming a neuron. Once it gets to its final resting place where it's going to live, it starts to grow those axons and dendrites. So with that, we can see around week 23, around week 25, we see that they're starting to reach out and try to make connections. So that doesn't mean necessarily that there's actually any kind of like conscious experience, but that these new neurons who have now moved to their final place are starting the process of reaching out, building axon branches, building dendrite branches, and saying, hey, what other neurons are around here? Let's start communicating. Let's start the process of doing brain stuff, which is pretty cool. With that being said, in this process, some of those stem cells are maybe more successful as they're becoming neurons, and some are less successful. So a ton of those millions of neurons that are being created do die off in this process. And so the remains of them are used as resources for creating new or to support the neurons that are surviving. And that's a big part of pruning is to use energy on some of the more efficient neurons. So basically the best functioning neurons survive. So stem cell divides, you've got, let's say both of those cells are moving to a similar place in the brain. You've migrated to your final resting place, let's say in the prefrontal cortex. One is a little bit better at making axon branches and dendrites than the other one. So we recycle the other one. We recycle the brain cell, that neuron that's not as good at making axon branches and dendrites because this one's just a little bit better. This one's just a little more efficient. So we'll focus on that one. So with this, these connections and neuron specification, that continuing neurogenesis and migration, that keeps happening up to about 2 years old. And then we see a big portion of what's happening in the brain development is actually pruning unneeded connections. So you've got the stem cells, they migrate to where they're going to live, they become neurons, they have axon branches and dendrites, they reach out to communicate, and then once that's all set up, once you've got kind of your whole brain trying to connect up with each other, you start to cut out and prune things that you're not starting to use. Because at this point, you're 2 years old, you've been using your eyes, you've been using your ears, you've been moving around, whether you're crawling, walking, right? By the age of 2, you're walking around, communicating with people, starting to learn language. So you're starting to use your brain, and you've had enough experiences now at this time that your brain's like, all right, these sections, these connections, these axon branches, these dendrites, we're not using them. Let's cut them. So let's be more efficient. Let's get rid of the stuff that we're not using. And so that's a big thing that happens across the lifespan. And it happens in a couple phases. It really waits to get started until everything's like set up. So you built more connections than you need. And then it says, okay, now that everything's built, now we can start being more efficient. Now we can start cutting things out. So if you look here, and this one is from your book, you can see the different stages of development or phases of development of these different things. So most of what we're talking about here is productions of synapses and neurotransmitters. So we'll look at these. These are a couple different areas. So the different dotted lines, hopefully you can see them on your screen. The one that prunes first here is the language and speech area of your brain, the one that lasts a little bit longer. So you're about a year old when it starts pruning, is the visual and auditory cortex. Again, you're already starting to use your eyes, you're starting to use your ears, you're hearing language, so you're focusing on those pieces that you need. And then the one that stays for longer, so up to closer to 3 years, is the prefrontal cortex. And so you start pruning things that you're not using there. In this whole process, there are other brain cells that we'll talk about in a minute, like glial cells doing myelination across your lifespan. And then in adolescence and then also into late adulthood, you also have more pruning happening as well. And so that essentially is the brain figuring out what does it actually need? What is useful? And after that first kind of big drop of pruning in your early childhood, so like before you're 5, before you're 6, before you're 7 or 8, after that, basically at that point, your brain is kind of done with massive pruning. It does go through a little bit of phase of massive pruning around puberty time, but mostly what you're doing at that point is maintaining and learning new things. So with plasticity, you can make new branches, make new connections, and then if there's things that you haven't been using for years, you remove those. So if you were playing guitar at 8 through 12 and then you haven't and you're now 38, you might have lost some of those connections for playing guitar. I can still play a couple of the scales, can't read music anymore, can play some chords, and that's about it. So a lot of those connections in my brain are gone. Maybe the next time I teach this class, I'll have practiced and rebuilt some of those connections. But that's the idea. That's what the brain is doing. You're starting with this neural tube. You are dividing that neural tube. About this point, you have some of those neurons to their final resting place. And as you're going through about 23 weeks, 25 weeks is when we see them kind of reaching out and trying to make connections. And so with that, you get up to young child, age 1, 2, 4, 6, depending on what part of the brain you're pruning connections. And then you have another big portion of pruning at adolescence. And by adulthood, you have kind of finished myelinating, and all the rest of the changes are, well, what are you using? Do you need to make more connections for learning new skills, making new memories, doing new hobbies? Or can we recycle some of these things, use a little less energy? Because as a reminder, the brain is using about 20% of your body's energy every day. So it wants to be efficient. And that is a big component of this whole process of how the neurons work, how all these processes in your brain work. So a lot of this is repeated, but hopefully you can see some of these connections with that we've talked about, and you can connect some of these pieces to things that we've seen in the rest of the lifespan. All right, so jumping ahead to other brain cells. We've alluded to these, we've talked about them in a little bit of detail other times, but there are other brain cells. And at the same time of the neural tube being created and making different cells, different kinds of stem cells can become different neuroglia. That's kind of our category. Glia cells or neuroglia are all the other brain cells that do things. And the more and more we investigate them, the more we are realizing they are a bigger portion of the neural mechanisms, like communicating, like neurons communicating with each other. So they're not just other things. It used to be that we thought, oh, they're just like the astrocytes and oligodendrocytes. We do myelination, and the astrocytes can help literally, neurons can like hang on to them and stay where they're going to live and help migrate with them to where they're going to live. But what we see is that they do seem to be playing a bigger role. Some of these help with the creation of things like central nervous system spinal fluid. That's what I'm trying to say. So the fluids in the brain, others bring nutrients to the neurons, take waste away, help with the recycling process. Some of these, it seems, are a big part of either creating neurotransmitters themselves to like package a bunch of neurotransmitters, give it to a neuron, or they help the neuron in its own creation of neurotransmitters. So they are a bigger part of the synaptic communication than we previously thought. And I think in the next years, we'll probably see more of that because there is more investigation into the neuroglia versus just the neurons. So here's from your book, basically what these different cells are doing. So maintaining neural networks, creating spinal fluid, supporting cells, and that can be things like recycling the waste. If a neuron dies or an axon branch, if we cut it off, we can recycle some of that components. This can be taking away some of the neurotransmitters in the synaptic cleft. It can be creating new neurotransmitters, giving nutrients to the cells, these kinds of things. So they're a big portion of the whole brain system. So when we're thinking of plasticity, this is really the main place that your book talks about it. We've talked about it quite a few times up to this point because it's basically the big portion of how your brain wires up. So the main idea with plasticity is neurons that fire together, wire together. And we see this in a couple different ways. The whole process of how plasticity works, how the neurons know to communicate in these networks, it's still not 100% understood, which is pretty cool. There's still things to learn about this. But long-term potentiation is one of the examples of how we know things happen. Basically, you've got glutamate receptors on on the cell that's receiving a message. So the postsynaptic neuron, so this is the one receiving a message, and this is the one sending a message. So there's already been a synapse, there's already been that electrochemical process, it's released some neurotransmitters, and here on the receiving end, on the postsynaptic neuron, it has some glutamate receptors. And essentially, if this neuron is sending a bunch of messages in pretty quick succession, this will start to put new receptors onto the postsynaptic neuron. It will say, hey, we need more receptors. Lots is happening here. Make more receptors. If there's more receptors to receive those neurotransmitters, the next signal is going to be easier. And so if we see a lot of quick succession of firing, basically the next neuron in the system is like, oh, hey, I need to make more receptors to receive this. I want to be efficient. So because I'm getting so many messages, I will open up more messaging notifications is kind of what you could think of it as. So if you've got email on your phone, text on your phone, some other messaging app on your phone, if you give all of those to one person and they really need to get a hold of you, they send you a text, an email, another message, you're getting all of those at once. You're like, oh, I need to do something. That's kind of what's happening with the postsynaptic neuron. It's getting so many signals that it's like, hey, I should probably put more receptors up here. Because I put those receptors up there, they stay there more long term. That's why it's called long-term potentiation. They stay there, and now it's much easier to activate because it knows, hey, I'm going to receive a message from this specific branch of an axon branch. So essentially, the cell is learning to fire in this sequence, in this pattern. It's learning that it's part of a circuit or network. And the main thing it knows molecularly is I'm getting messages from this specific space. I need to make it easier to receive messages from this specific space. So as we've talked about for memory, a memory circuit is like 1,000 neurons. You could think of that as one of the processes that's happening in a memory. So as your hippocampus helps rerun that memory program, like while you're asleep, you're getting more glutamate receptors. Make it easier for that. Right? And so this is how plasticity, one of the ways that plasticity works is this long-term potentiation. Put more receptors on the second cell, makes it easier to get a message and then send a message down to the next one in the row. And if that's happening to all of them in that network to say, play a guitar chord, well, that is now the network for playing that guitar chord. All right, another thing that we see is kind of the opposite of that, and this is called long-term depression, so LTD, and it's basically the inverse. And so we have those receptors here. Long-term depression is essentially, hey, I haven't been used for very long. This connection, I'm not really receiving messages from it anymore. That friend is not sending me messages or hasn't for a while. So, I'm going to remove them from my contacts list is kind of what's happening here. You literally remove that receptor from the postsynaptic neuron, and that is one way that we can prune that connection. Now, you can build up that connection again if you start getting messages. So using that guitar playing example again, if I start playing guitar again, there might be enough connections there still, and that axon branch and that dendrite branch might still be there in my brain that they can start working again and a little bit quicker. But another couple ways that we prune things is we can literally reabsorb or recycle or cut off and recycle the waste of those axon branches or those dendrite branches. So that's a little more extreme than this, but that's another example of plasticity. You can literally like cut those branches off if you haven't been using them for such a long time. The brain's like, well, the cells themselves are like, well, we don't need this branch. It's not being used. It's costing me energy. Get rid of it. Again, the individual neurons don't have like inside out characters in them. It's all just chemical processes. Uh, the cells themselves, they want to survive. They want to be able to do their job. And if they have extra waste that's not being used, they'll recycle that. So that is long-term potentiation, getting more receptors here to make that connection easier. So the next time you wake up and you try to remember that memory, well, you've got a couple more receptors there to do that. Or you have even more new connections. You can make new synapse branches and axon branches. So axon and dendrite branches to make more connections. And then if you're not doing something, you can lose those connections or you can lose those receptors so that network is not really active anymore. Okay, so with this also leads to the discussion of sensitive and critical periods. It seems that we've evolved the capacity to learn and develop certain skills at specific times. A sensitive period is what most things are, and this is the time in your lifespan when it's easier to learn a specific thing. Versus a critical period is essentially that you have to learn it now. If you don't learn it now, if you don't learn this thing or build this skill, you're maybe not going to be able to do it in the future. So I have a link here about sensitive periods in brain development. I have one image from the paper, but let's talk about it. So these are some examples, and this is a little bit older research, so a little bit newer research might suggest that these sensitive periods are a little bit broader than what we see here. And that's one thing that we saw. We used to use critical period as, oh, you have to build this skill at this time. As time has have gone on, we've seen more evidence that, okay, maybe it's more of a sensitive period. If you don't learn it in that span, you still can afterwards. Oh, and maybe sensitive periods are actually a little bit broader than we thought they were. So that's nice to see. It's kind of optimistic for development of these skills. But we can look at something like language in yellow. So this language one, it's pretty high in your first couple of years of life. By the time you're about 8, you should have hopefully learned language from the people around you, and we're really attuned to this in our first 3 years of life. Versus we can look at something like red here, looking at symbols or seeing symbols and reading and shapes and things like that. It has a shorter span of time there. And then we can look at something like hearing. And this makes sense for hearing because really you're pruning a lot of those early on in life. So in your first couple years, first year, first 2 years, you've had a lot of experience, hopefully, to hear different things, hear language, hear sounds, hear a cat meowing, hear a fan in your room. And so your brain's like, all right, we've got enough data now. Let's cut some of those connections. And this is one reason that these are sensitive periods because of brain plasticity. So some of this is synaptic pruning, leading to you not the brain not focusing energy on that specific area. But you can pause this really quickly and look at the different ways or the different periods for sensitive periods. But yeah, the sensitive period for language is basically before age 7. So if you have not learned a language by that time, it's a lot harder to learn a language. In addition to that, this is also related to multiple languages. If you learn 2 languages before you are age 7 or 8, you're going to be one much more fluent than someone who learns them later on, and also it's going to be easier for you to learn more languages too. So this is why it's called a sensitive period. In high school, in adulthood, you can learn an additional language. It's just a lot easier to do it because your brain has more of those connections still there. It hasn't pruned those connections away when you're younger. So this is why something like a dual immersion school, preschool, kindergarten, first couple grades of school are so useful because we don't see really any reduction in like reading, writing, um, arithmetic, so math. So we don't really see reductions of those scores, but we see a big jump in the next couple of years if those kids have 2 languages, both on math and on reading, and they have more than one language. So dual immersion schools very early is definitely better to get within that sensitive period. It's still possible, right? You can learn languages in high school, in adulthood. You could use Duolingo or something like that, but it's more difficult for your brain. You've got to build new connections versus they're there and they haven't been pruned yet early on. So a critical period, an example of that is the visual system. So if you're not using your visual system early on in life, like in your first year or two, you are going to prune a lot of visual system connections. Some people, especially people who have limited vision, we've seen that some parts of their brain in their occipital lobe actually take on and process sounds and other sensory information. So the brain's like, well, we're not really using this area. Let's use it for something else. So that's another example of plasticity. So that's what a critical period is, though. It's if you do not use this skill, if you do not gain this capacity, you might not be able to. But one thing that we're seeing is that it does seem with better investigation that things are more sensitive than critical periods. So most of what you'll see are discussions of sensitive periods, which I think is optimistic for our human brains. They're plastic enough that we can learn new things across lifespan. All right, so with this, we've alluded to this. I've talked about this quite a few times in class, but a big part of the synapses, of the sensitive periods, about learning, about building and breaking habits, it's really about energy, the energy efficiency of your brain. Your brain is trying to balance what will keep you alive, what will help you survive, what has worked before, with how much energy is it using. Because our brain uses about 20% of our body's energy. If you remember way back to one of the first chapters that we've covered, the resting membrane potential takes about, what was it? I can't remember if it was 3% or 3 total % of your daily body's energy, or it might be 3% of this 20%. I should probably look that up. But just having your neurons ready to fire does take energy. And so the body wants to be efficient. The brain is already using so much energy compared to the size that it is, that this pruning process, this reduction of synapses, this learning with building networks through plasticity, so having a habit, whether that's a bad habit like an addiction, or a good habit like I always work out this time of day, or I always drink water at these times of day, those kinds of habits are hard to break because you have neurons that are more efficient at sending those messages. And so hopefully now you understand a lot more about that and you can see those connections across the lifespan and have a lot more sense of, well, what goes into this whole developmental process from having a neural tube all the way to having a pretty much fully formed brain. Then it's getting myelinated, it's finished myelinated, but we're still using that energy efficient process. We want the brain to be doing what we want it to do, but also be using as little energy as it can. So that is chapter 7. The next piece we'll go into is epigenetics in chapter 9. Hello, students. This chapter 9 is planned to be in connection with chapter 7, but depending on the quarter, I may cut it into its own chapter, depending on how things have to go. Let's jump into it. Some basic principles here are to understand what exactly are we talking about when we're talking about a gene or genetics. So what is a chromosome? What's DNA? What are genes? Let's go over that. Hopefully you've had this in other biology classes too. Maybe it's way back in middle school, but hopefully this isn't brand new information. Here on the screen, hopefully you can see these on your own screen. We see a cell here and we have an example of a chromosome. And essentially what the chromosome is just a long, big wound up thing of your DNA. So this is the DNA in a strand. Here we've got another picture of a chromosome. So again, inside of the nucleus of all of your cells is all of your chromosomes. So this is a big wound up chromosome. We look here, it's all wound up, and we can see it is actually DNA. So what is DNA? The DNA is a complex molecule. It's just a really, 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 really long molecule. And that molecule, the order of the molecule, the ATCG, the genetic code, essentially tells your body what to do. So a gene is a unit of that hereditary information. It's essentially just one small piece of the DNA, or long piece, depends on the gene. It's the order of that, the ATCG. What is that order? It's a bit of that DNA strand that gives instructions for your body to do a variety of things. So proteins are the biggest thing here. So the DNA can basically instruct amino acids in your body and in those cells and in those cells to become proteins to go and do something in your body. Another thing that can happen is proteins can come and read that DNA strand, the code, the ATCG, and then go do something in your body. So a protein you can think of as just simply it's a bunch of amino acids collected together in a way that we call it a protein, and it regulates and directs body processes. These are all sorts of body processes. So how to a protein can direct how neurons can get their energy. A protein can direct how to color hair, right? There's a bunch of different kinds of proteins in your body. So here we've got a cell in your body. We've got in there some chromosomes. You can see those little X's. Here you can see, hopefully a little bit bigger in this picture, the wound up DNA. And if we look at it carefully, we basically call the different molecules. We've got adenine, cytosine, guanine, thymine. I might have got this pronunciation wrong, but that's the ATCG, and they just collect in a specific order. So, really long molecule, this is DNA, wound up together to be a chromosome. All those chromosomes are in your cell. So, oh, here we've got the ATCG. So you can see they're connected up. So adenine, cytosine, thymine, and guanine. Okay, I think I might have said one of those wrong, but that's okay. So essentially, here comes a protein, a bunch of amino acids that do stuff. It reads this DNA. It goes and does something in your cell. Maybe it makes a cell more to produce a color or change the color of a hair. Maybe it tells the neuron to, hey, use a little less energy, or hey, use a little more energy, right? So protein reads the DNA, goes and does something in your cell, and your body is made up of cells. So really, this is the main idea of genetics. Humans have about 20,000 protein producing genes. We have other genetic codes, so other sections of our DNA that say do stuff, but they're not active. For example, humans have the same genes for scales as some other creatures do, but those aren't active in humans. So it's kind of an evolutionary holdover that's just not active. And you'll see some of the process of activating and deactivating genes as we talk about epigenetics specifically. So the basic idea here is that you have in every one of your cells, you have DNA all wound up into chromosomes, and that proteins can come and read the order of the molecule on the DNA, go and do stuff in your body, and that's what makes your whole body. So genes work with other genes. Most things that are genetic are not just one gene. Most things are multiple genes. So eye color, for example. The reason it's not as simple as, say, Punnett squares as you might have learned about with the original identification of genetics, while it's not that like this size and this size or this color and equally matched, that's because most things in humans and other animals and plants take more than one gene. So genes work with other genes to do the process of telling your body to make black feathers, white feathers, yellow beak, gray hair, brown hair, to make the muscles, the muscle tissue or the muscles in, say, your arm, more muscle tissue, less muscle tissue, divide more, divide less, be more active, less active, all sorts of things. It's all directed by basically the order of those molecules in your DNA. So it directs the creation of proteins, and proteins do stuff in your body. So genes collaborate with each other to do things, but other things in your environment can affect those genes. So light can affect the hormones in your blood. The hormones get into your cells. They can turn genes off and on. So that's one example, say melatonin versus sunlight. So more sunlight, melatonin will drop. When it's starting to get dark, melatonin in your blood will go up, and that can affect what the cells are doing. So other things, stress, exercise, nutrition, radiation, temperature, sleep, they can all affect gene expression. And again, this is multiple genes at the same time. So while that's kind of a big, broad picture, that's really all I need you to know for this specific class. If you go more into neurobiology, you might go into a little more depth in, say, like grad school about this, but the basic idea of all of your cells have chromosomes. All of those chromosomes are basically made up of wind up DNA. The DNA gives your body instructions to do things through proteins. That's really what you need to know. So if you kind of understand that concept, or if it sounds familiar enough now, that's all you need to know. And we'll talk about how we study behavioral genetics. So in humans, the main things that we do to study behavioral genetics are twin studies and adoption studies. This is really the kind of work that we're doing to figure out, well, what is genetic versus what is the environment? So the nature-nurture question. And we look at behavioral genetics to try and calculate and figure out, well, how much of any given thing, whether it be schizophrenia, whether it be extroversion, whether it be eye color, how much of that comes from nature versus nurture. So for twin studies, with twin studies, we're talking about the similarities between identical twins, fraternal twins. We can, because identical twins are essentially like clones. So they're usually about that genetically similar. It's not a perfect similarity, just because biological processes oftentimes aren't absolutely perfect, but it's close enough. Even a direct clone can have some slight differences. But that being said, that's a little beyond the focus of this class. They're similar enough. They're very, very similar genetically. We can compare to fraternal twins, which are more similar genetically than just siblings. And so we can compare, well, are all these twins? So there's these twins. How many times do these twins show schizophrenia versus fraternal twins versus siblings versus parent and child to get a sense if the pieces that make up schizophrenia, how much of a genetic basis does that have? And that's kind of the idea here. Another thing that we do is adoption studies. This really helps us see both we can compare the birth parents to the adoptive parents, and we can look at the child who was adopted, and we can compare their traits. We can say, okay, they did or did not have schizophrenia, they do or do not have anxiety, and they were raised in this kind of environment, but their genetics are here. It really helps to kind of start to peel apart, well, what parts of their behavior or their traits are coming from genes versus the environment? How much of it is the nature part, the genetics, versus the nurture part, the environment around them? But it's a pretty complicated thing. We often, when we're doing this, looking at families, twins, genetic studies, adoption studies, we're trying to determine a ratio of what can we say this is kind of caused by the genetics versus this is how much of it is caused by nature. Putting numbers to that is what quantitative genetics is. So quantitative studies are number studies. You're putting numbers to something. So another example of something quantitative would be how many students raise their hands in class. I could count that. I could put a number to it versus a qualitative study you don't do. It wouldn't be the same for qualitative genetics, right? That I don't think that exists actually. Might be wrong. Qualitative would be something like, in my research, qualitative study would be like interviewing heroes, for example, asking them about their lives, getting that rich sources of data, even though I might not be able to put numbers to it. I might look for patterns and similarities, even if I'm not putting numbers to it. So quantitative genetics, you're trying to put a number to what ratio of this behavior or this trait, so aggressiveness or schizophrenia or anxiety, how much of that is due to the genetics versus the nurture piece. And these are expressed in numbers from 0 to 1, so the odds ratio. Sometimes it can be confusing interpreting odds ratio. It's not that they're really that difficult to understand, but honestly, at this point in your program and your education level, unless you're in a stats class, don't really worry too much about odds ratios. If you go into grad school and they're talking about odds ratios, you'll get a lot more of that. We don't really have time to go into odds ratio specifically, but I'll give you an example. So this is an idea of quantitative genetics. This specific study isn't talking about an odds ratio, but it gives you an idea of what are we trying to do with quantitative genetics by putting a number with how much of something is related to genes versus the environment. So this was a study just a couple years ago, 2019, and it essentially looked at people's reports of being LGBTQ plus and their genetics. And so it was trying to say, okay, we've had a couple individual studies. We want to take a bunch of data. So one of the samples was a UK biobank, also 23andMe, where they got a lot of their participants. So total, they had 477,500 individuals. And so reporting their sexual orientation and their sexual behavior and comparing their genetics. And so what we could see, we were basically looking for commonalities. Are there common genes expressed by these people, or are there common genetic markers that these people have that were, I think they focused on non-heterosexual. And then in the study themselves, they broke it up into lesbian, bisexual, gay. Um, so go ahead and read the paper. You've got the title here, you've got the authors here. You know how to find research. Go and find the research if you're interested. But the main idea is right here that the genetic variation was between 8 and 25%. So essentially, we could account for that behavior. We could predict it on 8 to 25%. So 8% to 25% of the variation that we see in sexual orientation is down to genetics. Genetics is about somewhere between 8 to 25% of the puzzle of sexual orientation. I just tried to give a couple unclear examples. I'm going to try to give a clearer example here. So let's take this high end, 25%. So we take 4 people. Let's say all 4 of those people have this same gene. Chances are one of those 4 people are going to be LGBTQ plus, right? So the genes aren't the full story. There's other pieces to the puzzle. And that's the idea behind quantitative genetics, is you use that to see how predictive it is. Certain things have more of a genetic component or less of a genetic component. But understanding every piece of the puzzle can help us, especially if you're trying to do something like treat schizophrenia. If it's, and I'm just making this number up, but if it's 98% genetic, well, then maybe there are things that we could do epigenetically or to help with some of those other biological processes. It's not that high. It is higher than this, like 25% genetic. But that is the idea behind quantitative genetics. So try and put a number on how much ratio is the genetics versus the environment. So hopefully that example was clear and helped you see it, even though this wasn't using an odds ratio. Again, worry about odds ratios when you get into grad school. It's not that complicated, but it's not a stats class. And so we're not going to go into that in a lot of detail. This is further complicated, this idea of trying to parse out how much is the nature versus the nurture, because our nature is also affecting the nurture practices. So we call these heredity environment correlations, so connections or relationships between things. So passive genotype environment correlations, evocative genotype environment correlations, and active genotype environment correlations, or niche picking, are some examples of one other way that this can kind of get muddied up and hard to parse out. This much versus this much is genes versus environment. So passive genotype environment correlations, I think this one, at least for me, is kind of the easiest to understand. Let's take this second picture here. Let's pretend that music ability is 100% genetic, or even like 75% genetic. It's just a funny example, but let's say having perfect pitch or knowing how to play music well, that is mostly genetic. Well, if you have both parents who have that gene, the kids are probably going to have this gene. So this parent has that gene, these kids have that gene, so all of them are probably more likely to do well at music. So what that means for these kids are they are probably already being raised in an environment, a passive genotype environment, that is supportive of that genetic trait. So the passive genotype environment correlation means that the environment is already supportive of their genetic predispositions. So the genetic predisposition is to be good at music. If the parents have that genetic trait, they'll probably have more music in the house. They'll probably have more instruments. They'll probably want to teach the kids to play music because it's something that they have. So the kids are passively just already, the kids themselves, or an organism is passive in an environment that already supports the genetic traits that they have. So I don't have that genetic trait, I don't think. Again, it's not actually a genetic trait, but it's just a funny example to make it more clear. So I am in a house that is already supportive of this genetic trait that I have. And that can make it even harder to say, okay, it's definitely the genes versus environment, because They're correlated, they're related. If you have that trait, if the parents have that trait, they might make an environment that's really good for that trait. Another quick example of a passive genotype environment correlation. Let's pretend that reading speed or reading comprehension is like 98% genetic. Again, it's not, it's just a funny example. If your parents have that genetic trait, they're probably going to have a bunch of books in the house. They're probably going to read to you. So you, as the organism, are in a passive environment. You are passively in an environment that already supports your genetic trait of reading quickly or reading comprehension. So that's where that passive genotype environment correlation comes from. The organism is in an environment. They are passively just going through it. They're not affecting the environment. The environment is already there to support their genetic traits. That's passive. Next one is evocative. This is you're essentially unintentionally creating an environment that supports your genetic traits. So pretty similar to the last one, but you are eliciting your behavior, your characteristics that come from genes elicit an environment that wouldn't be that same way. So let's pretend being friendly or unfriendly is 100% genetic. Again, it's not, it's just a funny example. We can even say 90% genetic. So if you're a friendly kid, you'll probably get more friends. People want to be friends with the friendly kid. Maybe you're better at social situations, understanding what the other people want to say, whatever it is, right? If you're a friendly kid, you will probably make an environment where you have more friends. If you don't have those genes, if you're not a friendly kid, you'll probably be in an environment, you'll create an environment where people aren't being your friend. So you'll get less practice, you will not make an environment or elicit, really elicit's the better word, or evoke. You will not evoke an environment that supports that trait because your genes aren't there. Right, so that is the evocative genotype environment correlation. And again, the main thing here is that makes it really hard to parse out how much of this is genetic versus how much of this is environment, because the genes have now elicited a new environment. The genes have created a new environment, not intentionally, right? The kid isn't trying to make an environment with more friends. It's just that's their traits. They're making an environment with more friends. Okay, the next one is a little bit different, but similar to the evocative. It's called the active genotype environment correlation, or niche picking. So this is essentially you have a genetic trait, and based on that trait, you seek out environments that are supportive of that genetic traits. So an outgoing kid, so let's say you are a friendly kid, you might actively seek out more friendships. So similar, but a little different. Or let's say you have really good body kinesthetic awareness. Right, there is some genetic component, but let's say it's like 70% of that is genetic. Again, just an example. But if you have really good body kinesthetic awareness, you'll probably pick like dance, you might do parkour, go to playgrounds like this. Like you'll actively pick activities that support the skills that you have. Right? It's kind of like a basketball player who genetically they're tall already. So they're really tall. Maybe they have got good hand-eye coordination. Some of that's genetic. So they've got those genetic traits, maybe a little more than one of their siblings. But then because of that, they actively pick, hey, I'll try out basketball. People have been saying that. I'll try it out. So now when they're now in the NBA, why is that? Is that their genetics or is that the environment? Well, it's obviously a combination of both. And that is why that's tough to get these clear pictures of this percent is genes, this percent is environment, because your genes will affect your environment. We change over time. We change the world around us. We evoke a world around us, or we actively pick specific situations. So as a quick reminder, passive genotype environment correlation, the organism is passively in an environment that just supports their traits. Evocative, they unintentionally create an environment that supports their genetic traits. And active genotype environment correlations, they actively pick an environment that is supportive of their genetic traits. So when we are thinking about quantitative genetics, trying to piece out that it's 25% this versus 75% this, it can be really hard because our genes are affecting the environment. And we'll talk in just a minute about epigenetics. And we've talked about it before in other chapters, depending on where I've put this in the class, that your epigenetics, you can have environment actually change some of those genetic factors as well. So with this, quantitative genetics is a really good area of study to try and figure out the puzzle of humanity and how we do things, why we do things, how our brains work, all sorts of things. But it is hampered, it's hard because of these things, right? Because in real humans, we can't ethically, obviously, we can't put an experiment and clone a couple people and see the different ways they're raised and how much of it is genetic versus the environment. So that would be unethical. We do it with rats all the time, though. So we have some ideas there. All right, so the next piece of this puzzle that makes quantitative genetics hard, but also really interesting, is epigenetics. So epigenetics is actually the environment impacting how those genes are read and expressed. So one thing to be really clear about here, we've heard a couple examples in different sections, whether it be on drugs, whether it be in things like stroke, whether it be on things like psychological disorders. There are some epigenetic connections of things like a baby inherits genes. They have exposure to prenatal toxins, maybe prenatal stress, um, maybe they have less nutrition, and that affects how their genes organize their body, organize the way that they process serotonin. So those genes activate or activate differently based on the environment that they're in. But we can see this across the lifespan as well. We can see the genes affecting we can see the environment affecting the genome across the lifespan. So as a reminder here, we have the chromosome. It's a big wound up piece of DNA, and I cut this picture for the earlier slides, but here what we're going to talk about is the histones and methylation patterns of DNA. Essentially, methyl groups and histone patterns are already there in your DNA. You've got them right now. These are some of the ways that your stem cells that we were talking about in chapter 7 with the developing into a neural stem cell. That's one of the ways that a stem cell knows to become a neural stem cell versus other stem cells that become other organs in your body is partially because of the methyl groups and histone patterns in your DNA. The histone patterns are, like we talked about, the wound up DNA being the chromosome. One of the things that it's wound up around are these histone proteins. So it can be wound up really tightly, it can wound up a little bit loosely, but histones are a piece of making that chromosome this shape that we call an X chromosome. So histones are a piece of the epigenetic puzzle, and the methyl groups are another piece. Methyl groups kind of say, don't read this next set of code. So we've got some ATCGs coming up. Don't read that, proteins. Doesn't matter. Not important to you. Maybe that's for the instructions for a skin cell, and you're actually a brain cell. So that is one way cells know, those stem cells know to become a neural cell, to become a bone cell, whatever it might be, is from these methyl groups and from these histone patterns. And those things, those methyl groups and those histone patterns can be affected by the environment. And that's really what epigenetics is about, is these epigenetic factors. So more stress can say, make this not as tightly wound. And so this piece right here that's pink, this gene, this set of code, the ATCGs right there, maybe before it was so tightly wound you literally couldn't read it. So a protein was coming on, it was like nothing to read here, I'll skip over this section. But here, because of some epigenetics, some stress or something like that, you have it looser wound. And so now the protein's basically looking around for something to recognize and do. It gets a message. It's like, I should do something in this cell. I should tell the cell to make less serotonin. Right? That's an example. And this is caused by this epigenetic factor. That can go away. Maybe you're not so stressed anymore. You've worked through trauma, so these epigenetic factors can be removed over the course of your lifespan. But the main thing here is we've got histone patterns to wind up the DNA or loosen up the DNA. Another way I like to think about this histone pattern of winding or not winding, let's say, let's use my fingers, I'll try to get them in camera. So I've got 4 fingers, let's say these are molecules. So we've got A, T, C, G. So maybe my protein that I want to read is looking for these 2 in the middle, right? So So T and C, it's looking for this instruction right here. If it's really tightly wound, let's say the protein is a molecule about this big, so it can kind of read two at the same time. So it's coming in, it's trying to read, it's like, oh, no, that's not the right one. Ah, there's the right one. Now I'll go do something in a cell. Versus if this is really tightly wound, it's possible for that protein to be like, okay, this looks right. And now I have a different instruction because I'm getting A, T, and C, right? Because that same protein, just the molecules, is reading that and it's like, oh, I've got a different instruction than it would have had otherwise if it was like this. So it goes and does something different. If it's really loosely wound, so here the molecules are further away from each other, it comes and reads and maybe it doesn't get an instruction. So it's like, I don't know what to do. I'm not finding instructions for myself. Or maybe it's like basing its instructions off of just this portion. It's like, okay, I'll go and do that. But that is a different instruction than if it got kind of what you would expect it to have. That's really what those histone patterns are doing, and the methyl groups are saying, don't read this next section at all. So hopefully that was understandable. I've got a good video in a second that I think will make it more understandable, but that process is essentially what we're looking at here. So the main idea is that the DNA code, the order, the ATCG, that's always the same. You clone a person, it's basically the same. So the ATCG, the order of that doesn't change. Right? So you're not actually changing the DNA. Something like radiation could actually change and mutate the DNA, but that's not what we're talking about. Epigenetics is not changing the code order. The code is still the same. It's just how much of that code are you reading? Are you reading a portion that you're supposed to be reading or that you're not supposed to be reading? That's really what epigenetics is about. So an example of this, one of these I think is discussed in your book, is the 5-HTT-LPR gene is related to a couple different things. We've talked about this before, depression. It is a gene related to the propensity for developing depression, but it only activates when a person has a stressful life. So take two people. Actually, let's take three people. I changed my mind. Let's take four people. All right, we got four people here. Two of these people over here, so closer to my pinky, these two people have the gene, the propensity for depression gene. These two people do not have the propensity for depression gene. So they're less likely on average to get depression. These two are more likely on average to get depression because they have this gene. Okay, now we go through life. Let's say these two people do not have a stressful life. Rock on. So these two people don't have a stressful life. Neither of them develop depression. Right? So these two people, they have a stressful life. Because this person has the gene for developing depression, they get depression with a stressful life versus this person doesn't. So that person, because of the gene and because of the environment, now some of those the proteins are reading the DNA in a specific way that makes it so it's way more likely for this person to get depression. So that person has depression. Half of these people had stressful life, and two of these people had the gene for it, but this person didn't have a stressful life, so they didn't get depression. This person had the gene for depression and had a stressful life, so lucky them, they've got depression. So another example of this is this gene does more than one thing. And that's one thing I want to be clear is that genes are complicated and work together with other genes. So there is a long version of the HTT-LPR gene that is protective against parent loss. Versus if you have the short version and you have some dramatic thing like parent loss happen to you, it is a risk factor. So the long version is a protective factor against really harmful things like losing your parents, versus the short version is not. So genes do more than one thing, but again, it's also epigenetic. So I think these examples, we may have seen them at some point in the class. I know I watched this in my lifespan development class, sometimes in general psych class if we have time. So I feel like we might have watched this video before. I know there's another video that we watch in the chapter on stress, specifically on epigenetics. So that's a really good one. Feel free to review that one if you didn't see it the first time. But let's watch this one from SciShow about epigenetics.
So say you have a clone or a secret identical twin. Because you know that a twin or a clone is a genetically identical person to you, which is crazy. But it's true. Anyway, so you have this clone or your twin or what have you, and your parents decide that it's too much trouble to keep both of you. And so they give your clone to a kind of sketchy traveling circus. Now I know your parents probably wouldn't actually do this, but just roll with me for a minute. So for the purposes of this example, let's just say that you grew up living a normal suburban American life, while your clone grew up with less stability and less access to nutritious food and less education, but more exercise and more access to sword swallowers and bearded ladies. And let's say you guys are 50 years old or whatever, and you get together and have some lunch. So you're sitting there across the table from your clone. What do you think you see? Well, for starters, Clone probably has a tattoo on its face. And if Clone had stayed in the circus, Clone probably, professionally rides a unicycle over a tightrope, so Clone is probably in pretty good shape. But then again, Clone probably smokes cigarettes and probably was malnourished as a child, so might be slightly shorter than you. By the same token, let's say that you've been eating Sonic tater tots 5 days a week since 1992, so you're looking a little bit tubby. Plus, you spent the last 20 years at a really stressful job that puts you at risk for all kinds of weird health complications. So, at 50, you and Clone would probably look pretty different from one another. But it turns out, you don't just look different, you actually have become different. So now if some scientists looked at your genetic code, they would say that your DNA was still exactly the same as that other person. That is to say, if your genome is a paragraph, all of the letters would be in the exact same order. But on another level, there would be a huge difference, and if we extend this metaphor, we can say that the letters are in the same order, but the spaces and the punctuation are all in different places. Of course, completely, potentially changing the message of that paragraph. The study...
So before it continues, I just want to make a point from this paragraph example. So I'll switch between the two. So here is the first way that the paragraph was, and there's the second way. So the punctuation is different here. Essentially, this metaphor is looking at the code. So again, the genetic code, the MYFAVORIT, like that word, my favorite, that's still the same in your genetic code. Epigenetics doesn't change the ATCG. It does not change the order of the code. It is changing the methylation and histone pattern so that you can read more or less or read differently the little ATCGs in the gene.
The study of this genetic punctuation that I'm talking about is called epigenetics, which literally means above genetics. The epigenome doesn't change your DNA, but it decides how much or whether some genes are expressed in different cells in your body. Epigenetics looks at what happens to your genes over the course of your life and whether those changes could be passed down to your children or even your grandchildren. So here's the way epigenetics works. You have billions of cells in your body and they each contain your DNA, the same exact blueprint of your genetic code. But just because they have the DNA doesn't mean that they know what to do with it. They need outside instruction from these little carbon and hydrogen compounds called methyl groups. The way these methyl groups control the genome is by binding to a gene and saying, do not express this gene. The methyl groups bind differently to your genome in a skin cell versus, say, a tongue cell or an eyeball cell. And that is one of the ways that a cell knows that, hey, I'm a skin cell, or hey, I'm an eyeball cell, or hey, I'm a muscle cell. In addition to methyl groups, epigenetics is also controlled by histones, which are proteins that are basically the spools that DNA winds itself around. Histones can change how tightly or loosely the DNA is wound around them. If they're more loosely wound, the genes can express more, and if they're more tightly wound, then they express less. So whereas the methyl groups are more like a switch, the histones are more like a knob. Every cell in your body has a distinct methylation and histone pattern. And that is what gives every cell its marching orders. Think of your genome, the DNA, as the actual hardware of the computer, while the epigenome is more like the software, which tells the hardware what to do. The genome is what's going to be doing all the work, but the epigenome is going to be telling it what to do. So the hardware of your DNA is going to be the same throughout your entire life. But these epigenetic tags do change throughout your life, and they decide what genes get expressed or not. Now, epigenetic information in a cell isn't permanent. It can change throughout your life, and it can be hereditary. And it can change over time, especially when your body is going through a lot of changes, like, say, during puberty. A bunch of methyl groups kick in and they're like, okay, so you guys over here, you're gonna have to start growing hair, and you guys over here, I really need you guys to get behind giving this guy some really horrible acne. Or like when you get pregnant, which hopefully I won't, but when you do, your epigenome has to be like, all right, guys, everything's about to get a whole lot bigger in here, and we're gonna have to pass something the size of like a Merriam-Webster's dictionary through that little pipe over there, so let's get this thing done. But it's not just these dramatic times when the epigenome is changing. It changes subtly throughout our entire lives. And it changes based on a lot of environmental factors, like what we do, what we eat, what we smoke, and how stressed out we are on a daily basis. Scientists have found that things like a bad diet can actually lead to methyl groups binding to the wrong place and making mistakes. And with those bad instructions, cells become abnormal and become a disease. And then basically all hell breaks loose and you get cancer or something. Epigenetics is a very young science, though we've known about the epigenome since the 1970s. It's only in the last 20 years that we've even known what effect these epigenetic tags are having on our DNA. And even after they got all that business straight, scientists still thought that all of our epigenetic tags were stripped off of our genome before they were passed on to our children. So if you started smoking 10 packs a day when you were 10 years old, that would certainly be a horrible health decision for you, but you wouldn't necessarily be harming your unborn children in any measurable way. However, the thinking on that is changing pretty rapidly. Because it's true that a lot, even most of the epigenetic information from a parent is stripped off of the embryo's genome in the first few days, and fresh ones are created specifically for this new person. However, some of these tags get stuck on the genome and are passed down from generation to generation. And it just so happens that the more they study this, the more it looks like bad epigenetic information is being passed from generation to generation. And this is a whole new way to think about how we pass information between generations. Your grandmother was making dietary decisions that affect you today. As we experience all of these new strange epidemics, diabetes, autoimmune disorders, cancers that weren't appearing in previous generations, it's starting to look like these may be caused by epigenetic information passed down from our parents. I know! It's such an unbelievable buzzkill! There is no point in our lives when we can do anything without guilt anymore. The discovery that the environmental factors a parent
Hopefully you can see this pretty well on the screen. We've got a couple epigenetic tags here: smoke, eat a lot, and the video is talking about how our environmental decisions, the environmental factors, can change those tags. So going back to our picture from our book, this is literally what those are. This is saying methyl group can bind to, maybe it's supposed to bind to here, maybe it binds to here instead, and so this whole bit of gene is not read. Maybe it doesn't bind, and so now this bit of gene is read by a protein. Also, here's an epigenetic tag, something that changes the molecules of the histones to be like, hey, I'm going to wind up more loosely. So more stress, less stress, smoking, not smoking, pollution in the air when you're in utero. Lots of environmental factors can change the patterns of these methyl groups, of these histones, so that your genes are expressing less. Again, the ATCG along this DNA is the same. It's the same in this chromosome versus the next chromosome, but they do change the methylation and histone pattern. So here we see the histones are very tightly wound up. In this case, in the example, the gene can't actually read this, or the protein can't actually read this gene at all. Versus when it's loosely wound, it can now read it. So now that trait is expressed, or the body is doing something differently. With that, another example could be, he mentioned that the histones are more like a knob. Maybe it's supposed to be at a specific place where it's not super tightly wound, so you get no message, but you get some message. So it's supposed to be a little loosely wound, and you might be able to read the ATC, versus if it's all the way loosely wound, well, now you're getting a very different message. So histone patterns are the winding and unwinding of the DNA into a chromosome. It can decrease it so tightly that the proteins literally can't access a piece of the gene to read it and then do something. Or it can be a little looser. Maybe that's what it's supposed to be and read some of that. Maybe if it's too loose, you literally have those DNA molecules further away from each other and you're getting a different message to that protein to go and do something else. So loose winding makes it easier to access, read, and express that gene. Too tight of winding makes it so you can't access and express the gene. So histones are like a light bulb knob where you can be totally off to all the way on and anywhere in between, versus methyl groups is saying do or do not read the gene. So it's like a light switch just going off, on, off, on. So let's look at one other example. We're not going to watch the whole video. I just want to point out a couple examples in this. It's a fun video using the Eevee, a Pokemon, to give an example of epigenetics. The whole video is pretty funny and fun. I'm only going to look at 2 specific examples it gives of epigenetics in animals.
Now, with animals, what epigenetic researchers have discovered is that some animals will be born with one set of genes, but potentially have parts of their DNA that can be unlocked by certain triggers in their environment. A great example of this are honeybees. Yeah, I know, I just dedicated an entire episode to bee talk as it relates to Ch7. Maybe I'm just going through bee movie meme withdrawals, I don't know. Bees are fascinating creatures, and in this case, they are the perfect Eevee example. So, like I covered back in that episode, female bee larvae can grow into either a queen bee or a worker bee. What makes the difference here is the food that they're given at birth. Bees given royal jelly become a queen, while everyone else becomes a worker. So in this case, royal jelly is basically the evolutionary stone for bees, determining what form the larval bee evolves into. It even changes the bee physically, where, in their adult stage, queen bees and worker bees look way different, but in their larval stages, they're practically identical. It's exactly like
I'm going to pull up this picture. It's talking about those larval bees being very similar. You could do this with 2 clones. You probably wouldn't for bees. You wouldn't take the money and effort to clone the bees. You would just have siblings. So that's what they did in reality. They've done this with sibling bees, but you could do this with clones. You could take 2 genetically identical bees, give one royal jelly, and it becomes a queen, and one you just give. Basically, bees eat like honey and starches and stuff like that. It becomes a worker. So these physical traits are based on the methyl groups, those pieces binding to the DNA saying do or do not express this gene. So it's that light switch example of turning it on and off. All right, let's look at another example that's a lot more like the histone patterns, winding and unwinding the spool of the DNA to read more, express more versus express less.
in their environment, the royal jelly, or lack thereof, led to wildly different gene expressions. And bees aren't the only place in nature where this happens. Another Eevee-like example happens in the agouti mouse, which has a gene which changes the fur of its coat. These two mice are genetically very similar at birth, but due to differences in nutrition, they have very different gene expressions, with one having a gene expression that causes it to become obese and have lighter fur. Same DNA sequence, different phenotypes, all due to environmental factors. In humans, it is.
I'm going to pause right here. So it's actually the agouti gene in these mice. I don't think they're actually called agouti mouse, but it's the agouti gene. The main point is you can see here, this one is a lot darker, closer to what we generally expect this mouse to look like. Over here, much more blonde, much more obese. This is an example of those histone patterns. The stress and the diet of this mouse is making their DNA spools unwind and express differently than this mouse. Again, these are sibling mice. They could be clones. Again, you could have clone mice. All of these could be genetically identical mice. In reality, they're just siblings, but you could do this with clone mice. They have the exact same genetic code. Just the diet alone can lead to this difference. And that is one of the cool things about biology, is we're seeing how many systems are interacting with each other all the time. So again, really quickly, the methyl groups, that's the on-off switch for express or do not express this gene. The histone pattern, or the winding up of that chromosome, can wind it up so much that it says you literally can't read this piece of DNA. You can't even access that ATCG. To being more and more loosely wound to express more and more, or express differently. It might not necessarily be expressing more. It can just express differently. Maybe you're getting more of the code than you expected to as the protein, and now you do something different in the body. So a good place to review might be these 2 videos that we watched in the chapter on stress as well, and how that can affect the immune system. But the idea here is, when we're looking at behavioral genetics, when we're looking at nature, nurture, when we're trying to figure out, well, how much comes from genes, how much comes from the environment, that having the heredity environment correlations, your genes can affect your environment, but your environment can also affect your genes. Your epigenetics can be changed by your environment. So that adds to the puzzle and the complexity of why brains do what they do, why bodies do what they do. And it's a pretty cool thing that I hope you have a better understanding of in this class as a whole, because this really it really connects well with developmental psychology. So, as a reminder from the beginning of class, I let you know this. I'm a developmental psychologist. I am really interested in changes over time. And these are some of the ways that our bodies change over time. Epigenetic changes, over time, the development of the brain. So these chapters 7 and 9 are some of my favorites, and I think really useful as both a beginning of class thing to look at and an end of class thing to look at, because it really helps put things in context that all of these pieces of the brain puzzle that we've been talking about, it's a bunch of systems. There's still more to learn about all of these systems. But I think one of the main things, and I'll describe developmental cascades, but I want to come to my final point first, and we'll come back to that. Um, we have the environment, we have genetics, we have the brain chemistry that you have the brain structure that you have the habits that you have. But that being said, because of plasticity, because of things like epigenetics, you can author a unique developmental path for yourself. You can change your environment, you can change your habits, you can change your networks in your brain, and this goes for everybody. So everybody, whether they are two and in a portion of sensitive period for learning language, or whether they are 92. You have the cognitive capacity, you have the plasticity. I mean, assuming at 92 you don't have a develop or a neurodegenerative disorder or something like that. Aside from that, though, that 2 year old, that 92-year old, your brain is plastic. You can learn, you can build new skills, you can build new networks. Yeah, it might be a little harder to do some new things for the 92-year-old versus the 2 year old, but all of us can, and this is kind of my main point that I want to come to, we can author our own unique developmental path for ourselves. So some of the things in the book talking about disorders or talking about the neurobiology of things, it might seem like, oh, these are just ABC causal factors. This is exactly how it's going to turn out. But no, there's a lot more variation, variety, change possible with brains. And I think that's a hopeful thing to share with people who you might be treating, that plasticity exists. You can change your environment and literally change how your genes are expressed. And these are some things that a lot of doctors and researchers, medical researchers, are looking at, or whether it be medicines or doing things like exercise, how that can help. So this model, I think I've shared it before, and I come to it a lot because I think it's a good visual model of things. You can have things that are maybe risk factors or protective factors. You might have an epigenetic factor that might make it more likely that maybe you suffer from anxiety, or you might have an epigenetic factor that makes it a little less, right? Having one that makes you suffer from anxiety, risk factor. Not suffer from anxiety, protective factor. So Plinko, this game, you drop a ball in here and it bounces around. It might bounce all the way over to here and then bounce all the way over to here. The goal in the game is to like earn the most money or like win the most money. But I think of it as like this is maybe risk factors or negative outcomes, and this is positive outcomes. Maybe the positive outcome you're looking at is expand your working memory a bit. Maybe the positive outcome is break a habit, right? So those are the kinds of positive outcomes we're talking about. And that's the developmental cascade model. This is the same over here in this picture, this cascading waterfall. You've got maybe negative outcomes over here, positive outcomes over here. Genetically, epigenetically, the environment you're born into, maybe you're right here at the beginning of your life. Right? So maybe you have additional risk factors and that water droplet brings itself over here. Then you have some protective factors that build up over here. Maybe you make some choices, go to therapy or something like that. It's over here. Maybe something bad happens, some big stressful event. Your water droplet comes over here. But at any point in time, this water droplet or that aspect, that trait, that behavior, that habit has the capacity for some movement and shift. And little movements back here might lead to many years later a really positive outcome. So that's one reason I really like this developmental cascades model, is because it really goes to show with plasticity, with the kind of change that's possible in your brain, we can have positive outcomes over the lifespan. So these complex behaviors, these genes, these disorders, traumatic brain injuries, whatever it be that we talked about in this whole class, they might push you one way or another in the developmental cascades. But with this, you can change environments to be a protective factor. You can make changes in your life, whether it be exercise or working on breaking that habit, going to therapy. There's a lot of things we can do epigenetically, but there's also things that our brain is doing on its own that we can maximize the potential of that plasticity of our brain to look for positive outcomes. So that's why I say one of my main points here, while we've learned a lot of detail about how different processes work in your brain and your body, we as people can choose to author a unique path for ourselves and change our environment and change our brains, because that's what's happening all through the lifespan is change. So I'm a developmental psychologist. That's why I bring all my classes back to development, because it really is all development. So thanks for being part of these chapters. Again, right now when I'm recording this, this is the last chapter of the class. In the future, this might not be the last chapter of the class. It might be earlier on in the quarter. But thank you for being with me in this class, whether this is the end or whether this is closer to the beginning of your course. I hope you enjoyed learning these things. Email me some questions, and hopefully you are really interested in learning more about the brain, whether the class is beginning or ending.